Hello, everyone.
Hello, good morning. Good afternoon.
Yeah, good to see you and welcome. Okay. So let's just give 2 minutes for the other people to join. I don't know if there are some people who are still on Teams. Yeah. Yeah, I think some people are still stuck here. They are not seeing my messages. Okay, Chris. Okay, welcome everyone. Chris is hosting the meeting today. I'm handing over to you, Chris. I'll go to Teams to try to get the other people.
Perfect, Dorcas. Come back with a lot of participants.
Okay.
Welcome everyone. First of all, hi David. Do you want to share your screen? Good to see you. Good to hear you. How are you doing?
Yeah. Good. Greetings from Iringa, Tanzania. You want me to share my screen now?
Yes, please. Let's check if that works smoothly, and I'll give a brief introduction when everything is working fine.
Okay. Can we see?
Okay, great. Yeah, we can see it. Can you start the presentation mode? Nice, perfect. All good. Okay, thank you. So again, welcome everyone. As Dorcas said, I'm hosting the session today. So let's first start with the usual housekeeping. First of all, we always try to keep the invitation list for this meeting updated. And well, sometimes new people join in the CG and in your program. Sometimes mail addresses change, and now some people just slip through the net. So please, if you are aware of someone who should join and is not on the list so far, or well, you know that you're not on the list, which probably means you're not here, which is a bit useless, I guess. Well, anyways, if you know someone who's not on the list, please forward the invitation or just let us know that we should add someone, because we want to make sure that everyone is included, everyone can participate, and everyone is up to date. Secondly, this is a safe space to present and to discuss ideas. please keep an open mind. I know it's important to be critical. It's important to ask critical questions, but please do that in a respectful way. And well, just make sure that we have a great and good community here to share ideas and to discuss ideas. So now it is my great pleasure to introduce David to you. I met David when I visited Tishale from Seat Beans earlier this year in Tanzania. And David gave Jeff Ehlers and me back then a short tour in the field and told us about the key features about the Artemis project he's going to talk about. It's a digital phenotyping project, and first of all, I was very skeptical because I had seen a lot of stuff about drones and the likes, which, well, people have nice projects off, and I've never seen anything working in practice. It's too complicated. I don't know, there's too much data generated, people don't know what to do with it. But, well, David managed to convince me. It is a really nice, simple and powerful technology, and the data output he showed me was really impressive. So I thought this would be a great topic for you here to know about. And with that, I don't want to spoil the whole story and pass it on to you, David. So welcome again, and looking forward to getting your update.
Yeah, thanks a lot, Christian, and thanks for the invitation to present at this meeting. It's really a pleasure. Yeah, so my name is David, and I'm at the Alliance, working on the digital inclusion research level based out of Arusha, Tanzania. And my principal responsibilities at this moment is continuing to develop and execute on Artemis. So Artemis is, as Christian was saying, it's kind of a labor of love that I've been involved with off and on for maybe 3 or 4 years now. And it's involving a lot of really diverse partnerships. So it's funded by the Gates Foundation, as Christian was saying. It's developing phenotyping tools, digital phenotyping tools. We're partnering, of course, with IITA, Colorado State, London School of Economics, and then Mineral. So Mineral is a kind of a bit of a wild card for us as the CG centers. They are an Alphabet company. So this is Alphabet is the parent company of Google. And so Mineral falls within Google Alphabet, relationships of companies. And so we're able to draw and to leverage basically the best in-class imaging, AI, and data systems engineers in the world, and apply those really transformational talents and skill sets to some really hard but very important problems, which is how do we develop better varieties of plants faster and more locally adapted. So let's see. Yeah, so Artemis, in the first phase anyways, we're focusing on a couple of commodities. The first one is common beans, which we've been working on for a couple of years now. We're working on cowpeas and sorghum. And so the idea here is that cowpeas and common bean are relatively similar plants from a morphological perspective. So if we can develop some imagery tools for one and transfer it to the other, that would be a good leverage for learning that we can maybe move across the grain legumes. And then sorghum is just very different from either of them. So if we can develop good imaging tools and technologies for sorghum, then we can say, okay, maybe we can start working on some of the grasses in addition to some of the small legumes. So the crux of the problem that we're trying to solve for is that, phenotyping in its classic sense and essence starts from human observation, right? So we have breeders and technicians go to the field. They have years and years, decades of training, and they use their eyes and a notebook and experience in order to make qualitative estimates of what they're seeing on the field. So we know that this is, can be very powerful, but it's also difficult to scale. It takes a breeder, decades of experience in order to get the right type of intuition in order to make these estimates. And there can be, differences in error depending upon the time of the day, depending upon if the breeder is sick. We need to use technician over, throughput, moving technicians through the field, and also technicians coming and going. So there's a lot of sort of uncertainty and error that's associated with some of these qualitative estimates that we're getting. And of course, that leads to error in our decision making. So the concept here was to leverage deep learning technology. So essentially looking at machine observation. So machines can observe and record physical structures and more in the environment and make quantitative estimates, quantitative counts that humans just can't do. So this is the same technology that's being used to drive AI in self-driving cars, facial recognition on some smartphones. And so it's the same technology, and how can we then apply this to do phenotyping to be able to assess sort of plant parts, in this case, pod counts, as you're seeing in the background. So our initial target technology was a rover. This is a mineral rover, the one that you're seeing there on the bottom right-hand side. And the idea was, can we start with something really powerful that can leverage multiple different cameras at high resolution, so we can produce, really accurate 3D models, and we can do this across fields and generate terabytes and terabytes and terabytes of data at really minuscule levels, for key commodities. And so we initially started with 2 rovers at CIMMYT. So one was for wheat and one for maize, and this is working with Matt Reynolds and his group, and the other one was with beans, so working with Michael Sevelraj and his group with beans. So, we quickly realized, and this is kind of really well known, and I think Christian mentioned this, a lot, is that there's a lot of weaknesses and sort of over promises with some of these, high technology devices. One is that they're extremely expensive, right? One's going to be $250,000 plus. So that just means that the specialized maintenance is going to be required, and it's not scalable, right? If we wanted to do this, let's say at research stations at CIMMYT's HQ in Mexico, CIAT's HQ in Cali, okay, then maybe we have enough infrastructure and tech expertise to do that. But a lot of the really important breeding is happening in these research stations. So, Arusha, Tanzania, or Lilongwe, Malawi, or Eastern DRC Congo. And so these technologies are just not scalable. We can't put them there really under any circumstance for any reasons. So really, these are transitional technologies that we're using to develop our initial models. And then from there, we really wanted to know, can we do this for mobile phones? Because we know that mobile phones are really ubiquitous. They were really the number one target that we had as a project. They're affordable, they're scalable, right? Just about any breeding station I've ever been to, doesn't matter where, doesn't matter what country, doesn't matter when, most of the breeders and technicians have smartphones. So this is a tool that's already existing in someone's pocket. And instead of moving from $250,000, we're moving to something in the order of a couple of $100. So this is something that we really wanted to target, and that development of newer models of handsets are just getting better every day. So at some point, there's going to be conservative momentum where we're going to have really high resolution phones in everyone's pockets all around the world. So the weakness is at the moment, there's really quite a variable handset specifications. We have these really inexpensive Huawei and Tecno phones coming from China that cost less than $100. And on the other end, we have, a Samsung Galaxy S23 Ultra, which costs thousands of plus dollars. And so there's a big spectrum there. So how do we know which ones will be able to really create good models if we can at all? I think the other thing that we're trying to solve for is that throughput efficiency. So, getting a rover through a field, you just press go, the rover moves. It takes a lot of deployment really quickly. A drone is even faster. A human eye is relatively fast. But a mobile phone, how high of a throughput can we possibly expect from a mobile phone? And these are questions that we didn't know and we wanted to investigate a little bit more. So we developed this mobile phone annotation and model development pipeline. And we've been working on this over the past year or so. And so essentially what we've been doing is working with technicians in the field using mobile phones to develop standard operating procedures. And so this is, how do we actually take a picture? What angle we take a picture? How many pictures do we take for each individual trait that we want to work with? Do we need multiple images? Do we need panorama images? Do we do it in the morning? Do we do it in the afternoon? So there's a whole range of sort of challenges that we have to kind of simultaneously solve for in order to develop good SOPs, in order to develop good models. So once we've developed the SOPs, then we can move to training data. So now the the images are now in the cloud, and we can then do some hand labeling and outlining. In this case, this is bean pods. So labeling the bean pods, and it's actually cowpea pods. So labeling the pods in order to train some of the models. Once we have good enough trained labeled data, then we can start using models and developing models that then can do trait extraction. So then we execute these models, we take the traits of interest, we convert images to things like pod count or plant stand or flower count. And so more or less, this is sort of the pipeline that we're doing at the moment in order to generate the models from which then we can deploy, further down into the future. And so some initial results that we have about a year into the project for pod counts with common beans, we're finding that we're able to produce Pearson correlation R-squares of around 0.84 from about 500 images. So this is kind of in the range that we're hoping to get, but we're anticipating maybe having thousands of images that were required. And given really good SOPs, we can nail this down to 500. So, much less than several thousands of images to produce a really good quality pod count model. And for cowpea, we're developing Pearson correlation R-squares of 0.83 from about 600 images. And so this is helping us to assess some of our initial hypotheses that if we can do for one type of plant, like a legume, a ground legume, a bushy ground legume, then can we then transfer those learnings, and will it apply also to cowpeas, a different type of ground legume? And so far we're finding out that yes, we can. If you can develop one for one, then more or less we can develop similar scores of accuracy for another one, given that the SOP development can happen in an efficient and precision type of way. So for stand counts with common beans, we're getting Pearson correlations of 0.94, and we're able to make these models from as few as 100 images. So really very, very few amount of images. And we were thinking about having an order of magnitude more images initially. So this is one of the outcomes that we're getting so far that's very positive for us, that this might have a technology might have a future in actual deployment in the next couple of years. For sorghum, a little less R-squared, so 0.85, but from 500 images. So sorghum is a little more tricky, right? The initial seedlings are much smaller, harder to see. And so we need much more training data in order to do that, but still a lot less than many thousands of images. And so now we're saying, okay, actually we can differentiate and develop significant models between types of plants, between legumes and between grasses. So we're already starting to generate a lot of evidence that that's answering some of our initial hypotheses that we had as a project. So the next bit was scalable execution. So when we first started this developing the SOPs, it was taking us about 3 hours to image maybe 30 or so plots with several technicians. And so this was certainly not scalable. What this was useful for is helping us to understand what type of image quality we would need in order to develop really good models. And so once we had that, then we needed to think, okay, then how can we then take these images at scale in order to enable us to really start doing some relatively good throughput for imaging. And so we started working with really simple sort of structures. This one here was developed by a local craftsman that works on the side of the road using simple bars and bikes and welding. And we're able to then develop phone mounts that for pod count, we're able to collect 100 images in about 2 minutes. So basically one entire breeding plot, we can image every single plant, every single pod within 2 minutes. So this is something that we felt was certainly equal to, what the visual estimation was able to do. Right before I was coming down here in Riga, when we're still at the office in Arusha, where we're looking at stand counts. So we have a different mount for stand counts, and we're able to image 6 rows of plants. And so this has ended up being 180 plants in 30 seconds. Basically 1 pass, 2 phones to the field. We can do this in 30 seconds. And so this is really allowing us to give us confidence that this technology can be deployable. We can produce good models, we can get good results, and we can do that with relatively high throughput efficiency that would be useful for a breeding program. And so one of the things I keep telling our team all the time is, we don't want to use anything you can order on Amazon, because you can't order Amazon, let's say, in Eastern DRC. Right? We don't want to have any really fancy thermal cameras or anything that costs a lot of money that's been stuck in you have to fork out lots of money for it and quickly go obsolete. So everything that you have to do, you can be able to buy in a local shop in Eastern Congo that you can use to manufacture a bicycle, fix a microwave, or fix a really simple car. So only really pieces that you can use really anywhere in the world. And so that's what we're really focusing on. So then the pipeline development that we're currently working on, as I mentioned before, was field data collection, model development, and then trait extraction. And the bit that we're really relying on Mineral, the Alphabet company, for is something that we're calling a breeder sandbox. So at the moment, our field teams are collecting data. The data is being sent into the cloud. We have very expensive mineral engineers working on developing AI models. And then we're using this as an output for our breeding partners. And so what we're working with them on is thinking about ways that we can functionally move the AI modeling away from very expensive engineers sitting in California to more affordable data scientists and analysts within the CGIAR who are sitting in Cali, Colombia, or Arusha, Tanzania, or Lilongwe, Malawi. And so one of the core outputs of the project is to be able to have this breeder sandbox, to be able to say, okay, if there's a common bean breeder or a cowpea or a groundnut breeder in Malawi, here are some ways that you can collect the imagery data from the field, developing the SOPs. We can work with your data science in order to develop your models, and then you can develop the trait extraction on your own. So the whole pipeline could be really owned and operated by the people on the ground who are actually looking at doing imagery-based phenotyping. So what our vision is for the final integration is we want these tools to be able to fit, basically plug and play with what is currently happening now on the ground. So we're focusing at the moment not on trait exploration, but trait replacement. So we're not looking to measure the angle of the third flag leaf of the third plant in the plot. We're looking at, okay, what are breeders doing? They're measuring stand count, they're measuring pod count, they're measuring flower count, they're doing disease score. These are the things that they're working on. So these are the things we're trying to solve for. So data collection, a breeder would go to the field or a technician would go to the field. They'd collect the data that's already been used, that we have an existing model. They collect the data is uploaded into the cloud. The analysis happens in the cloud in near real time. The outputs then go into your analytical system of choice. Now, if it's enterprise breeding system, if it's whatever you're using, we will be able to develop a system that you can just plug and play. So it's just one tool that fits within your existing set of toolkits if you want. So an example, this is something that I showed to Christian, was an example of some of the data ingestion and analytical tools that we have. I'll just give a quick demo. It's called Field Browser. Just give me a second to load it up. Browser, okay. Okay, so you should be able to see Field Browser right now.
Yep, we can see it.
Excellent. Okay, so this is coming from the CIAT Alliance office in Cali, and so this is one of their plots. So we'll click on block 6, and we'll click the heat map. And so this came from them, I think this is past season. And so the way the outputs would be is we can look at the different traits that we have. Plant count per meter, canopy coverage, plant height, flower count, pod count. So let's get pod count. And then we can look at the data that's coming over different times of the year. So here's March, here's one in April, here's one in May. And over the season, each one of these cubes here, or these squares, represents a genotype. And these are genotypes within the entire block. And so I can click on, let's say, this genotype here, and it'll produce the plant count, the canopy cover, plant height, flower count, pod count per meter, squared for the entire plot for the entire year. So we're really getting quantitative, continuous data across all of our plots in a block. And then it gets you the outputs that you would get at the very end. So we can basically take the data, ingest, we can summarize it, we can produce kind of just generalized breeding statistics, heritability, genetic gain, whatever you want, or we can provide just the raw data and you can do also whatever you want, however you want. And if you don't believe it, you can look at the gallery over the seasons and you can see how then the plants and genotypes have been changed as it kind of moved the field. And so if you needed to say, hey, this is really weird, I don't know what's happening here, you can retrieve the imagery archive, you can go through it and you can triage to see, what exactly was happening there if you didn't have a chance to go to the field. Okay, so that is Field Browser, which is our current user interface that we're working on from the user side. Back to the presentation. So the next steps. So really what we want to do is we have really good information that we think we're comfortable moving. Basically, can we do this for every crop within the CG, every location, and have this really be a central part of the phenotyping platform? Yeah.
Have you opened the presentation again? Because I can still only see field browser.
Okay, let me change then the share. Okay. Okay,
Yep.
Christian,
There we are.
Okay.
Yeah.
Great. So the next steps, we got good confidence. Can we use this system and develop it to have all of the crops across the CGIAR, across all the locations, and have it be really a core phenotyping platform that can be integrated into the CGIAR breeding program? This is something that we think potentially could be feasible in the next couple of years, given existing technology. So we've been currently developing the next round of proposals from the Gates Foundation based on the outputs that we have so far. They feel very comfortable with us moving forward with a scale. So we want to look at small grains and legumes. So basically all the small grains and legumes, we want to be able to then do what I mentioned to you before, collect the data, develop the SOPs, develop the models, do trait extraction, and produce a fully scalable, deployable digital imagery-based phenotyping platform for small grains and legumes over the next 4 to 5 years. We have foundation commitment to be able to do that. The next one is an R&D in parallel. Roots, tubers, and bananas. We'd love to see if this is possible. We don't know. We've never done any digital phenotyping with mobile phones for RTB. Can we do it? If we can, what's the tool? What's the leverage that we're going to need? Maybe we can't, but we can see at least if we kai through an R&D. The next one is analytics. Now that we have these data coming in, continuous data from thousands of locations all over the world, what can we do with it? How can we use that to transform some of the breeding informatics? Can we further develop sort of the pathology toolkit to say, okay, pathologists are looking at sort of single plants, single genotypes on single plants and single pots in the greenhouse in order to understand the relationship, the genetic relationship between the particular strain of whatever pathogen and then the germplasm. And then they follow that through from greenhouse to plot to field. And so can we develop a pathology toolkit that can really be used across the entire chain of the breeding process? And then the thing that I'm most excited about that can be potentially most transformational is if we can develop these tools and technologies that work on the research station that are deployable on a mobile phone, then can we use those mobile phone deployment to start pushing breeding from on-farm, from on station to on farm? And if that's the case, then it potentially opens up a lot of opportunities to develop sort of in situ or in parallel to some of the research stations, varieties that are going to be locally adapted to local soils, to intercropping. So we can really look at G by E by M really on a large scale, and then really, I think, transform sort of how breeding is done just in general. So that's the potential, but there's a lot of risks that we can get there. But again, we have really good support from the foundation to give this a try. And so then maybe that's the last slide that I had. So a pitch to you would be great to hear input on what I presented to you. And if you want to be involved in the next phase of this project, now's the time. Shoot me an email, shoot me a message. We can start integrating your program into our proposals and start doing some digital phenotyping wherever you are and whatever you're working on. So another ask from me, I'm going to leave this on, if you don't mind, Christian. We are doing some sort of assessments of sort of needs and sort of desires from the different breeding partners. So we have a quick online form, and maybe I'll leave this open now. And if you want to take a look at it, maybe you can do that scan it via a QR code, or you can just go to that website and enter that code in. And there's like 3 or 4 questions that really help us as we're building out this program to see if we can really understand your needs and your wants for some sort of mobile phone-based digital phenotyping tool. All right, so Christian, that's it for me. Maybe over to you.
Perfect, thank you so much. So first of all, feel free to forward this to Dorcas and me, and we can spread it across the participant if someone, wants to do it later after the meeting. Again, thanks a lot for presenting this. As I said, I think this is potentially really powerful technology because of its simplicity. I'm sure there are a lot of questions. And also, well, as you mentioned, I think for everyone who's potentially going to work with this may be the opportunity to, well, put up requests or recommendations or whatever you want to say. So please, hands up or just jump in if you have questions.
Yes.
Yes, please, Jerome.
Hello, everyone. Hello, David. So I have a question regarding sorghum. You didn't present much about it. What's the status of advancement?
David, you're
We
muted.
cannot hear you.
Yeah, the sorghum group that we're working with, this is Jeff Morris's group at Colorado State University, and then Siras in Senegal. You know, it would have been ideal to have, you know, CIMMYT be involved, but when we were writing the proposal, there was that awkward transition with Equisat and CIMMYT. So sorghum, we were able to do plant stand really efficiently, and then, you know, there's a very short window for doing sorghum in Senegal and Colorado, and so we're going to be moving to doing sort of more of like the grain and sort of the reproductive and more into the vegetative stages this next season. So basically starting at the end of this month. But from what we've been seeing in the field, you know, imagery, if you can get a good image, you can build a good model. And really the trick has been, how do you get the good image? And sorghum, the tricky thing about sorghum, of course, is that it can be, you know, 2-3 meters high, and the spacing can be really, really kind of condensed. And so being able to get an image from that would be tricky. Basically, we ruled out, you can't do it from the rover that we had because the sorghum's too high. But I'm hopeful that we can be able to do that and execute that with mobile phone, using that sort of that simple push card. If we can, the way that it's working now with beans anyways, is as you're moving through the field, there are bars that are sort of opening up the canopy. You plant the phone sort of right in the middle where the canopy is open. It takes an image, and then as it's moving through, the canopy closes again. So then for me, I'm just thinking about if we can then move it through the field. If it's a sorghum plant, maybe the little cart needs to be a little bit more robust, a little bit heavier, a little thicker. So as you push it through, it can really move those plants aside. And then we can put as many phones up and down the line as you want to capture from the floor all the way to the ceiling of sorghum. So this is still like a thought that we have, but I'm pretty hopeful that if we work together and we really solve what is really the barrier to producing good images, then the models will come.
Thanks. Just following your question, so I was wondering for rice, even for beans, if you are taking the picture from the top but at a low level, would it work also?
Yeah, that's a good question. Rice is tricky because it's hard to get like it's hard to move physically through a rice field. You can take top to bottom, but you can also get top to bottom with like a drone or something higher. I think what's really useful about the mobile phone is that you can get underneath the canopy, right? If you need to look at things within or below the canopy, diseases or pods hanging down, I think this is where mobile phones have a little more versatility to be able to do that. And then of course we can put up a bar and put some phones on the top to do top down as well. But for me, it's the sweet spot is that under canopy, mid canopy pieces.
Thank you very much. Very interesting presentation.
Perfect, thank you. There's a comment from Randy that I'm going to read, and then we have a question from Gary and Elisa. So Randall says, looks very exciting, and I really like the more practical approach of using equipment that is available everywhere. However, your timescale still seems too fast for wide applications, as we need to expect a range of technical problems along the way. Well, need to find a few strongly motivated breeders to help move the system. Randy, if you want to move in, I think it would be interesting for David if you might share what you think are potential technical problems that might come up, and that, well, again, may help David and the team to be prepared for these.
I can't really say what they are, but I mean, I worked in private industry as we went with drones and so on, and we just came up was really exciting. They're using a lot of them. I'm out of the industry now, but we suffered quite a bit early on, even with high quality drones and so on, of really getting the data that we wanted in various technical problems of processing the images and so on.
Yeah, that's a really good, Ruby, very important points. Very good questions to answer. We can develop good models, right? I'm pretty confident now, having spent, a year in the field working with the teams, that the problems that we have to develop good models and deploy at a research station, they're not insurmountable, right? It takes a little bit of kind of like elbow grease and ingenuity, thinking about what can be deployed again in like Eastern DRC. And I think, Randy, to your point, and I agree with you 100%, is that when you move from a couple of research stations, a couple of research stations scattered around Africa to, thousands of stations across the world, to, tens of thousands of breeding plots across farmers' fields, then the challenges become exponentially harder and also different. Then this becomes like a data systems challenge, which is super tricky. It also becomes sort of like a logistics challenge about, what kind of phones are available? How do we make these machines or these little phenotype cards? Do we need to have one for every different type of plant, different traits? There's a lot of complexities. And that's really like going to be the meat and potatoes of this next phase of the project, right? This first phase was 2 years. Is there evidence that this will work? Okay, there is, yes. Okay, so the next phase is how can we make it work? And that's going to be really, really hard. So I'm confident we can make the models, and I'm hopeful that we can make it work. But in order to make it work, we really need to have really good input from people who are working on the ground, working on breeding stations. They can help us answer these practical questions. We've got, Alphabet as the technology company. So as far as like processing images, data systems, we're working with literally like one of the best organizations in the entire world to be able to do that. I think the harder part is how, as an organization of the CGIAR, do we make it deployable and functionable across all the diversity of locations and personalities and breeding programs. But so that's why I'm here. We really need everyone's input and collaboration on this.
I think maybe the closer analogy might be, we had cameras on combines. I was a maize breeder, and we're trying to do plant height and things like that. And I mean, you get different light, different times of day, reflecting, hitting the camera the wrong way. You get dust on it, and it just to get some accuracy on the different conditions, that seems like a simple thing to do, but it wasn't. And again, I'm just talking about some height measurement, simple things, lodging scores of the plants falling down. It took a while, and actually, as I was leaving 5 years ago, it was still marginally successful. Though the error rate for individual people, you have to compare it to what is the error rate for a technician going out there measuring plant height, for example, and that can be pretty high too. So if you can equal the quality of the technician and do it faster, then that's great. And I think you can get there.
Yeah, this is also a good point. So there's different types of algorithms that we're working on, right? There's object detection, there's segmentation, and the hardest one is going to be sort of 3D modeling. And at this point, 3D modeling is really difficult. Like plant height is super tricky. For things like counts, stand counts, pod count, disease score, those are pretty, I think, straightforward. But with anything that you need 3D modeling, you need to have either like stereoscopic phones, so having two phones that you can mount, and you need to have like standardized heights. So those are very tricky, and that's certainly going to be in the research part, but they're going to be tricky for sure, without doubt.
Thanks, Remy. Thanks, David. Gary, next, please.
Hi, thanks for an interesting presentation, and I missed the first 6 or 7 minutes. So if what I'm asking was covered, I apologize, and you can tell me fast. But just sort of following up on Randy's point, the use case in in commercial breeding was to reduce, partly at any rate, to reduce labor technicians actually taking ratings in large plantings, big areas. Where the use case in on-farm testing, like 1,000 farms, or in the genomics on-farm genomic selection project, is a small plot, 3 to 5 entries, and 95% of the time and cost involved in managing those small plots is getting to them. So the large savings in labor probably won't form much of the advantage. So the advantage of using images to capture traits in that context will pretty much completely have to come from increased accuracy. And so the control, or the baseline for accuracy, is a visual rating from a trained rater or a trained measurer, I would think. So is that, and they can be really accurate, and obviously they're technically easier to deploy. So is that the baseline that you're using, for stand count or for pod count, the repeatability of the and correlation with direct measures with a visual rating from a trained observer?
Yeah, so we have actually 4 different metrics that we're using for measuring accuracy. So one of them is we have a breeder and technicians in the field doing their phenotyping. And so that's sort of like one thing that we're looking at measuring against. The other one is we know that there's going to be error in that. Even the best trained technicians, at the best they can do qualitative assessments that's generally in the right region. So what we're doing is we're having plots that are small enough that we can do full quantitative ground truth. So we have people in the field every day counting flowers, how many flowers are open every day in the field, in every plot, how many pods are there every day in the field in every plot. So we can compare then what the models are being predicted are, and against what breeders are doing, and also against what ground truth, true ground truth is. So we can have a really objective comparison of, are we in the range of so yes, we have that data. And Gary, I'll be sending this in the report to Neil, and so he can pass that information on to you, when I submit it. Also, you should know, Gary, that I was 2 days ago, me and a bunch of colleagues of mine working on the project were in the genomic selection beans fields in southern Tanzania. And the way that they're set up, 3 really small plots would be really easy to image. And a colleague of mine, Bertha, who's working on sort of the user design bits, was working with sort of the technicians on the genomic selection to see how they would interact with it, how would they use it. And so we think that what we've seen so far with genomic selection should be a very easy use case for us to execute.
That's great.
Okay, thank you. So Elisa, next, please. And then we have Dorcas, Bodo, and Belko.
David, thank you very much. I am Elisa, potato breeder at SIP. My question is if this software is open source and it's possible to collaborate with different kinds of image, because we usually use we evaluate the flowering, senescence, number of tuber at harvest. That this is spent a lot of time under field condition for a breeder. So this is very amazing, this tool, because we support us and save time under field condition and standardize the procedure. So it's possible to start a collaboration with the potato breeders in order to standardize these tools.
Yeah. Yes. In phase 2, we have a research component that's dedicated to RTB, because we don't know. We have a good experience with sort of the small grains and legumes, but RTBs we don't know. So we would like to. So I put my information, my WhatsApp, and also my email in the chat here, and also Christian can pass this around. And let's jump on a call. Maybe we can arrange, I think there's someone else talking about potatoes too, Mokhtar Kante. So let's get like an RTB meeting set up that we can discuss how we should do this. And the foundation, they want us, they said, write the proposal and we'll see about getting you guys funding. So let's write the proposal. Let's see what you need or your needs are. We can put it together and see about getting a really nice RTB research project for Artemis in the next phase.
Okay, thank you very much.
Yeah, Chris, mine is not a question.
Yeah.
I just wanted to draw
Okay.
your attention that there is a comment in the chat for David from Kevin, which I think we skipped, just for your information.
Okay, well, let me quickly search for that comment. Sorry, here we got it. Sorry, Kevin. Well, David, Kevin said, Jeff Morris and ISRA sorghum colleagues are working closely with our CIMIT team. We will be happy to participate along with them. So, well, maybe it would be good to make contact between you two guys to figure out the sorghum situation that has been raised before.
Yeah, well, Kevin, it would be great. Maybe if you and I can also jump on a call. We'd like to do groundnuts, millets, and sorghums, right, which are all now within your portfolio. So that's going to be a major component of what we're doing. And I would say, like, I'll be back. I'm in the field right now doing some field visits. I'll be back on the week of the 21st of May. And from there on, why don't you just like send me a meeting invite and we can chat and we can think about how we can then put together the proposal for the portfolio of crops that you have.
That'd be great. Thanks. And good to see you active, David, since you left here. Great
Kevin.
to see you landing and doing great stuff. So thanks for the presentation. And yeah, it'd be great to work with you on this project, however we can.
Fantastic. We'll be reaching out very soon, for sure.
Thanks.
Good. Thanks again, Dorcas, for mentioning that. And sorry, Kevin, that slipped through the net. Now there's already discussion going on between Bodo and Gary. So Bodo, please jump in and maybe make your point yourself.
Okay, thank you. Can you hear me?
Yep.
Can you hear me?
Yeah.
Yeah, so actually, it's not me who raised his hand, it is Belko Nuhun of Africa Rice. But I'll take the opportunity as you asked me. So my feeling is, I've seen some of these developments, even in the private industry, but also in academia. For example, there was a student at Cornell University who worked for 3 or 4 or 5 years, and they ended up getting, I don't know, a plethora of data. And when they somehow had to stop or they shifted it to something else because we ran out of space, simple storage space for all the data they needed from a single field. And they worked amazed. And they said, they also said that this would need more time and more development. It is really high-end research. And then in practice, I came across, let's say, a more simple application in the past where a device would be used to count the tubers or to make an estimate of the number of potato tubers which were laid open in a heap in a plot, and then at the same time the average size and volume of each tuber was estimated by the same procedure, and then by a simple multiplication of the volume per tuber and laying this around with the specific weight, and then multiplying by the acreage, we came up with the estimated yield per hectare, which is a very useful information for the breeder. And normally nowadays all these roots and tubers and things need to be weighed. This is heavy work, and sometimes people would be happy to have a replacement of that. So maybe this is a suggestion where you guys could make relatively quick progress and come up with something that is very handy and useful in the hands of these people who are dealing with that. And the other things are, in my eyes, a little bit academic, aren't they? Thank you.
Yeah, no problem. Yeah, thanks. Thanks for the comment. On the data volume, when we had the rover moving through the field, it was something like 1 terabyte per week, which was just, it was enormous. Fortunately, we're not doing rover anymore. The images that we have, we're doing compression on them in order to put them through the cloud. And so they end up being in the kilobytes range, not in the megabytes range, which is good because that means we can use maybe smaller or cheaper handsets. It also reduces sort of data volume. But if we do this, if we have the vision that we have about really this becomes like the main phenotyping platform, then it's going to be a lot of data. Fortunately, we've already been talking with Khulud and Brian King and David Abreu and Sharifa and Younghwa about what kind of data systems do we need to be able to hold and handle all of this. Because also from, the perspective of the organization and also the foundation, we want this to be in the public domain. All these images have a lot of uses, maybe beyond just what we're doing, and we want that to be able to be available for people to experiment and do, new science. So within this first, the focal point of what we're doing now is really looking at replacement traits. So what breeders are doing now, and can we do them, digitally? But with that breeder sandbox, to your point, the breeder sandbox, we want to be able to create an open source imagery modeling tool that anybody can use for any purpose. And so then rather than like getting an email, hey, David, can you help us doing this, it's rather than, hey, here's like the breeder sandbox, you can use it to like develop your own models for, tubers sitting on the ground or like bean color or, whatever you think you can use images for. We want to be able to provide basically the infrastructure to enable you all on the field to solve the problems that you have that are unique to your situation, your breeding program, even your ideas. So that's really want to go is to really democratize this process rather than having it kind of be narrowed just to our groups and what we're doing.
Thank you. So, well, I thought you were going to ask your first question. Anyways, let's go with Belko first, and then I'm gonna get back to the other question in the chat. Belko, please.
Yeah. Thank you, David and Christian, for the presentation. Yeah, I was having the same questions with Bodo, the data management and so, but the other technical issue that I would like to know if you have think about or worked with that is when you work with crops like cowpea and you are using these bike type mounted phones, and I am sure you know about plant architecture. Some plants are erected while some are prostrated on the ground. So how are we working with this imagery system to take care of pots that are on the ground, under the leaves or above the leaves, either on erect plant or prostrated plants? So this is to do with grain legumes. And also I would like to know if you have a project, we are thinking of a project on rice in different ecologies. I mean, rice in upland and where this system can be used, especially at vegetative stage to do plant count, area cover, early vigor with competitiveness. These are traits that upland breeders are interested into, but I believe the system cannot, the way it is now, go into the mud in the lowland with water, and also giving the size of breeding field trials. For example, we have trials of 100 plots, trials of 1,000 plots. So if we are spending 2 minutes with one system per plot, it means it will take 3 hours for 100 plots and 30 or 35 hours for 1,000 plots. So it means we need to have different bike and different technician, operating them simultaneously to maintain the throughput. Beside that, yes, we will be interested to start and work and see how it can be applicable, especially in the upland. Beside that, at Africa Rice, we are developing and implementing a drone-based imagery system for high throughput phenotyping using RGB and multi-spec. So I would like to know if your system, your platform can also deal with images, RGB and multi-spec images taken above ground at 150 meters, and taking care of plant height, biomass, panicles per plant, like predicting yield. This is where seriously the bottlenecks are with what we are doing, and we will be happy to work with you to see if we can push it in that way for data management, data processing, data analysis, threat extraction. We don't need to work on thousands of threats, but the key ones that will help breeders to make their line advancement also. So thank you very much.
No, thank you for the questions. Very good points. Yeah, regarding prostrate plants, this is going to be hard. We have the same trouble with sort of the type 2, 3, 4, 5 common beans, especially sort of the intermediate, climbers. They're not full climbers, but they're not fully bush, and they kind of the little, they kind of connect with each other. So these are tricky, and some things may just not be able to then be imaged very well, and that's just sort of the reality. But some things can, and I think if we can really focus on what is it exactly that the breeders are looking for, if they need prostrate plants, if they need these kind of like these semi-climbers, can we image with them? And if we can't image it, how is the process by which we image him? This is like an iterative process, right? So every time we find a challenge, we need to work over and over and over and over again within the cycle, within the breeders, to see and answer if it can or if it can't be done. So I would say we don't know, but that's what sort of the next part of the project phase 2 would be, researching to see if it can be done. So the throughput, So if you really want a high throughput, if you want quantitative high throughput, then certainly smartphones are not the answer, right? You're going to need to have drone. If you quantitative data, then the best next bet is going to be the smartphone. And so maybe you mentioned bicycles. It'd be interesting to see, can you move the plots open to get higher throughput? What is the throughput that you need to make it work? Can the smartphone provide, that service to you? So these are good questions. And I would say like the smartphone platform is going to be one of the tools in the breeder's toolkit, right? Like if you have thousands of plots, maybe drone is your solution. And maybe a subset of that you want to do with the smartphones in order to get it through. Maybe some smaller stations, if they have really like maybe a couple of 100 plots, then maybe that's smartphone only would be the way to go. So these are things that the whole system, I think Randy's question before, doing it across the whole system as an executable, deployable phenotyping model, there's a lot of finessing that needs to happen, and that's really based on what breeders' needs are and what tools are available. Multi-spec, that's a good question. Can we integrate that into the breeder's sandbox? I think it should be. It's supposed to be robust enough. So if you put in images, basically you put in images, you do your own labeling, and then you have a platform you can do your modeling, and you can generate your own outputs. So let me bring that up with Mineral. I haven't chatted with them about that directly because we've been mostly working with RGB. I'll chat with them about that, maybe get back to you.
Good. Thank you very much. So what is interesting is if RGBs and multi-spec images collected by drone, I mean thousands of images, can be processed and analyzed, and also come up with threat extraction with some great R values that what you showed at 80%, 85% will be great.
Sure.
Thank you very much.
Thank you.
Yeah, so we are open to work with you and test it in the upland at least and see how it goes. So it's a good option.
Sounds good. I'll reach out.
Thank you.
Perfect. Thank you, David. Thank you everyone for the questions. We're running out of time, so we need to make that a bit of a fast end here. David, is there anything you want to say at the end? And then Dorcas, maybe if you have any final comments, and then we can call it a day here.
I think I've said enough. It's time for me to be quiet.
Perfect.
Yeah, thank you, Chris. Thank you, David, for the nice presentation and for accepting to present today. Just two things. First, I apologize for the confusion with the Teams and Zoom. So I would like to send out a new link just for Zoom. I would be canceling these occurrences, but replacing with a new link. So the occurrences and the frequency will remain the same and the time. And secondly, I'm looking forward to meeting you here in a month. The Genetic Innovation Quantitative Genetics team would be talking about selection indices. We will be sending out a reminder of the agenda 2 days before. This is a topic of interest for most breeding programs. And so please forward the link to all team members who might be interested in that topic. Thank you all and see you.
Thank you all. See you soon.
Thank you, Chris.
Very much.
Bye.